My name is Swetha Palaniappan, and I'm a junior at Cape Elizabeth High School. So for my project this year, I looked at the effect of microplastics on mammalian cell culture. So my project this year was really based off of my project from last year. So last year, I looked at the number of microplastics along southern Maine's coast. For my project, I went to 15 different beaches and collected ocean samples and looked at the number of microplastics in those samples. And my results showed that there was microplastics in every single beach that I went to. And in some of the beaches, the numbers were incredibly high. So I really wanted to see if these microplastics had any effect on humans and animals on the cellular level. I had read some previous research stating that microplastics have been found to affect marine organisms' respiratory system, that it's been found in human stool samples and fish brains, but there wasn't much research on how it affected cells, so that's kind of something that I wanted to look more into. So for my project, I used two different cell lines. I used MDCK cells, which are canine kidney cells, and L929 cells, which are mouse fibroblast cells. And I looked at how these cells were affected by exposure to plastics. So for my project, I chose two different types of plastics, polystyrene, which is commonly found in plastic bags and plastic water bottles, and polyethylene, which is commonly found in plastic food containers. In addition, I looked at how these plastics at different concentrations. So I chose three different concentrations based off of some previous literature. So I chose 1 microgram per mil, 10 microgram per mil, and 20 microgram per mil. And I looked at two different time points, so a 6-hour time point and a 24-hour time point in my project. And I also observed sort of 4 different aspects of change. So first, I looked at changes in cell proliferation using a hemocytometer, and then I looked at changes in metabolic activity using MTT assay, and then I also looked at changes in molecular markers using qPCR, and I chose 3 molecular markers. I chose superoxide dismutase, which is an oxidative stress marker, and tumor necrosis factor alpha and interleukin-1 beta, which are both inflammatory markers. And I looked at how those were affected by the exposure to plastics. And the fourth aspect of change that I looked at was sort of changes in the proteins. And to do that, I used SDS-PAGE. However, I could not present the results today because there was a chromosomal smear in the gel, which prevented me from looking at the data. So as for my results, for cell proliferation, as in the figure number 2, cell proliferation with exposure to plastics, there was a general trend of a decrease in cell proliferation with exposure to plastics. And looking at figure 3, which is the MTT assay, for the most part, it remained constant. However, there was, under certain conditions, significant decreases in the metabolic activity. And then looking at figure number 4, so starting with the first panel, which is the interleukin-1 beta, there is a significant downregulations under almost all of the conditions. With the superoxide the tumor necrosis factor alpha, which is the second panel, for almost all of the conditions, TNF alpha has been upregulated with exposure to plastics. And with superoxide dismutase, it's the same. Under almost all of the conditions, it has been upregulated with exposure to plastics. And so from this data, I was able to see sort of the negative impact that microplastics had on humans, looking at the cell proliferation, depending on the type of cell, a decrease in cell proliferation could affect the survival of an organism, and same with metabolic activity. And looking at the markers, the upregulation in tumor necrosis factor alpha shows that it could possibly mean that these microplastics are being viewed as causing inflammatory stress, and with the superoxide dismutase, it means that the cells are under oxidative stress. So overall, I was able to see that these plastics had a negative impact on the cells. And in the future, something I would like to do is I would like to see sort of the synergistic effect of using both the polystyrene and the polyethylene, and possibly using microplastics from the ocean and seeing how those affect the cells.
Thank you. Now we're gonna have some time for questions. So whoever would like to ask a question, go for it.
I have a question for you. Why did you choose these cell lines, the kidney cell line and the mouse fibroblast cell line?
So originally I had wanted to actually use human HEK293 cells. That's the original one that I wanted to use, but since I'm a high school student, there was a lot of additional training involved. So I ended up, I was able to use K9 kidney cells, which were the next available, and they're very similar to the human cells, which is why I went with the K9 kidney cells. And the L929 cells were, I really wanted to do another cell line, and the L929 cells were something that my mentor had available, which is.
Okay, makes sense. One last quick question is, is there a methodology that you're aware of that would allow you to assay or measure the plastic uptake in the cells?
Not that I'm aware of, no, but that is something I would want to look into.
Yeah.
I would be interested to hear about how you prepared the different wells. Did you add the cells first and then load them with the plastic? And did you load big chunks of plastic, small little particulates? What did that look like, and how did you think it affected your results?
So for my project, so first, when I got the cells, they were in one flask, so I had to like wait a little bit and let them multiply first. And then I moved them into the wells. And with the plastics, what I did was I combined them with the media. So I used, originally, when I first did it, the plastics didn't mix in well. So I had to add, according to the information that I was given by the company, I added some tween 20 into the media to help the plastics better mix in. So adding that, I added those to, I added that mixed in with the media into the wells with the pop.
Were these adherent cell lines? Did they stick to the plastic, or were they non-adherent? Did they float?
These cell lines were, I believe these cell lines were adherent, yes.
Did you do the cell culture in these assays?
I did. So I had a mentor, Dr. Bo Rostama, who helped me throughout this project. So he sort of, he showed me how to do it. And then after viewing him do it once, then I proceeded to follow after him. He sort of helped me through the entire process.
Gotcha.
Yeah, I was looking at the cell culture technique and whatnot, and I didn't see any wells or tables that didn't have the plastic particles in them as a control. However, since you're seeing a decrease in interleukin-1, I think that should serve as your control fairly well. Now, were the particles, plastic pieces you added, were they sterile?
Yes, they were.
And did you buy them sterile, or did you sterilize them?
I bought them sterile. So there was a company called Cospheric, and they were selling polyethylene microspheres and polystyrene microspheres that were sterile.
Good.
And for my control, I had a separate control welds. And for that one, I just used, instead of plastics, I just did the media combined with the tween 20.
Excellent. Okay. Yeah, no, the reason I think the interleukin-1 is important is because that usually comes in response to bacterial endotoxin. And so if you have contamination of your plastics, it will show up that way. So the fact that it went down is, I think they're pyrogen free, so that's nice.